लिंक है आपके पास? सर लिंक है आपके पास? नहीं, लिंक का देना पड़ेगा सर। व्हाट्सएप नंबर एक मिनट मैं आपको बताता हूँ। हेलो, हाँ लिंक आपको भेज दूँ क्या? हाँ ठीक। हाँ हाँ अभी कर रहा हूँ, हाँ यहाँ पे कर दीजिए 98738 488 47। डॉक्टर जावेद। हाँ भेज दें। हाँ आ गया क्या? ठीक है भेज दें। नाम ठीक है। Hello, sir. Hello everyone. So today's practical is on direct demonstration of bacteria by staining and the common media that is used in a laboratory. So for demonstration of bacteria by staining, first of all we have to make a smear. So for making a smear we require a glass slide, okay, which is you can see in the picture. This is the glass slide, and you have to put a drop of normal saline, either from you can put a drop of normal saline through the loop and put a culture from the colony, or you can directly use the broth and make a smear like this and let it air dry for few minutes Okay, so after air drying you have to heat fix it. Okay, so basically whenever you're preparing a smear you also have to make a control, prepare a control. So a known gram positive, a gram negative, and in the center you will put the test strain, whichever strain you are testing. Okay, and then after that, after air drying we have to heat fix it. Okay, so there are different methods of fixing the organism. So the main purpose is to fix the organism on the slide, and plus also fixation causes, it retains the morphology of the organism, so we have to fix it before we uh, before starting the stain. So we'll fix it first. So there are chemical methods and physical methods. So we are using physical methods. So chemical methods basically are most commonly used in where you have to see a internal structure, like in case of infected RBC, in case of malaria, where you want to see the internal structure of RBC, internal structure of the RBC and the parasite within it. So for that we have to make a chemical fixation because it retains the internal structure. So in that case we can use methanol or butyraldehyde or like that. So here we are using heat fixation. So here it fixes the organism and also inactivates the enzyme, thereby retains the morphology of it. So next, after heat fixing it, we'll do stain. So there are different methods. There are simple stain and there are differential stain. Okay, and there are negative staining and like that. So here we are using gram stain. So gram stain is basically a differential stain. So with the gram stain we are differentiating whether the organism is a gram negative or gram positive. Okay, so for gram staining, first of all what we'll do, we'll use a primary stain and then mordant, and then we'll decolorize it and then secondary stains. Okay, so the gram positive organism will retain the primary stain and gram negative organism will take the color of the secondary stain. Okay, so initially what we'll do after heat fixing it, we'll put the primary stain, which is a basically pararosylene dye, which is a basic dye. Okay, so in this case here is gentian violet. We can also use basic fuchsin, diluted basic fuchsin, methylene blue, and after pouring off the primary stain over the slide, okay, we'll keep it for 1 minute. After 1 minute we'll wash it under the tap water. After washing it we'll put the mordant, that is iodine. Mordant means dye fixator. So basically it fixes the dye. Okay, it forms a dye iodine complex. This dye, primary dye, and this iodine forms a complex which does not then come out of the bacterial cell wall in case of gram positive organism. So after that, important step, decolorizer. So we can use acid alcohol, acetone, or alcohol acetone. We can use, here we are using acetone. Okay, so acetone is used for decolorizing. So acetone, after the mordant, you're done with the mordant, you'll wash it. After that we'll put the decolorizer. Okay, so decolorizer is kept for 2 to 3 seconds. So this part is very important, decolorizer, because if you over decolorize it, all the organisms will look like a gram negative organism, and if you will under decolorize it, all the organism will look like gram positive organism. So this step is very important. So how do we know that it is decolorized? So you have to keep the decolorizer only for 1 to 2 seconds, or just keep the slide in a slant position and allow the decolorizer to run through the slide. Okay, it is done just for 2 to 3 seconds. After that you will wash it, immediately wash it with water, and then you will put the secondary stain, that is safraning. Okay, so gram positive organism will retain the primary stain, and gram negative will take the secondary stain, which is safraning and appears pink or red in color. So the basic principle behind this is There are many theories like pH theory, where we say that the gram negative organism, gram positive organism has a more acidic cytoplasm, so it will take the basic stain more strongly and for a longer time. And in case of gram negative organism, it is less acidic than that of the gram positive organism. And the another important theory is the cell wall theory, where we see that where basically the gram positive organism has a thick peptidoglycan layer as compared to the gram negative organism. Okay, so they are closely cross-linked tightly, and this forms a barrier, and through which the through which this primary stain does not come out. And on that also, plus this acetone or alcohol you're using, it what it causes shrinks or tightens this cross linkage of peptidoglycan. So it has a thick gram positive organism, thick peptidoglycan layer, about 50 to 100 layers, and the gram negative organism has a very thin peptidoglycan layer. So the diiodine complex easily comes out of it, and plus it also has a lipid layer, so which gets disrupted while using the alcohol and forms pores from which the primary dye can come out. So they take up the secondary stain. So in gram stain, we are differentiating bacteria into gram positive and gram negative. Okay, so another differential stain is Albert stain. Here we are staining the Corynebacterium diphtheria. Okay, so this organism basically has a metachromatic granule, which is do not confuse this organism with the spore. Okay, this is not spore, this is basically the metachromatic granule, which is a polyphosphate inorganic complex polyphosphate granules and some enzymes also, and they are called metachromatic because they show metachromasia. When this is stained with a primary stain like methylene blue, it does not, the metachromatic granule does not take up the blue stain, rather it takes up purple red color. This is because of the property of its metachromasia. So in Albert stain, these are the composition: Albert stain 1 and Albert stain 2. It is used to differentiate Corynebacterium diphtheria from other organisms. So we have two solutions: Albert stain 1 solution, Albert stain 2 solution. So Albert stain 1 has, we have two basic dyes, that is toluene blue and malachite green. Okay, and we have to for pH we are using glacial acetic acid and alcohol and distilled water. And Albert 2 stain solution has the modern, that is iodine, potassium iodide in distilled water. Okay, so like earlier I've told you to be making the smear. Similarly, in that way we'll make a smear, air dry it and fix it by heat. Then after that we'll put the Albert stain 1. Okay, will consist of the dyes and allow it to act for 5 to 7 minutes. We'll keep it for 5 to 7 minutes. After 7 minutes we'll pour off the stain. Okay, we do not wash it after Albert stain 1. We'll just pour off the stain. After that we allow to stand the slide on the rack, and over that we'll put the Albert stain 2, that is the mordant, and allow it to act for 3 minutes. After that we'll wash the slide and we'll blot dry it and we'll look it under the microscope. So what we'll see is, we'll see an organism with bluish. In case of Corynebacterium diphtheria, we see a greenish cytoplasm. The cytoplasm of the bacteria will be greenish. That means the bacteria will be green, and the metachromatic granules will appear as purplish red color. Okay, so basically this the stain which we are using, that is the toluene blue and malachite green, they both are basic stain. Okay, and the pH of the stain is made about 2.8. okay, that is acidic basically, but the metachromatic granules, they are very highly acidic, so this stain becomes basic for them. So the metachromatic granules take up the toluene blue and appears as purple red in color, and the malachite green is taken up by the cytoplasm and appears as green. So this is about the Albert stain. So next is Nielsen staining. Nielsen staining or acid fasting. Okay, so here we are staining the structure which are acid fast. This is basically used for Mycobacterium tuberculosis. Okay, the acid fastness is because of the presence of mycolic acid in the cell wall and cross-linking of fatty acids. That is basically cross-linking of fatty acids called mycolic acid. Okay, so in this, how do we prepare this? First of all, we make a this prepare a smear from the organism. Basically, it should be done in a biosafety cabinet because it will be aerosolized and infective to the infective to us. So it should be whenever making the smear, we should always make it in a biosafety cabinet because it is highly pathogenic. So how do we make it? After preparing the smear, as I've told you, we'll flood it with the primary stain, that is the carbol stain. Okay, after putting the stain, flooding it with the carbol 1% carbol is used. So we will heat it. Okay, we'll keep the smear on a rack and then we'll heat the stain, okay, until the vapors comes out of the stain. Or what we can do is, another option is, we can use a preheated carbol and keep it for around 5 minutes. Okay, after keeping it for 5 minutes, we will decolorize it. The decolorized used is either acid alcohol, or we can also use sulfuric acid. So in case of Mycobacterium tuberculosis, we can use 25% sulfuric acid because it is acid fast, highly acid fast. And for Mycobacterium leprae, we can use 5% sulfuric acid. So in this case, we are using 3% HCl in absolute ethyl alcohol. Okay, so we'll keep it for around 1 to 2 minutes. Decolorizer is kept for 1 to 2 minutes. After keeping 1 to 2 minutes, we'll wash it and then we'll stain it the secondary stain, that is methylene blue. Okay, so we'll keep this secondary stain for 30 to 30 seconds to 1 minute. After the time is over, we will wash it. Okay, we will wash it and see it under the oil immersion field. So what we'll see is a Mycobacterium will look like pinkish organism, thin, slender, long, pink organism with beading appearance. See, there is beaded. It looks like a bead. Okay, so they look like beaded organism in the stain. Okay, so this is basically used for staining the acid fast organism. The whole of the background and the other organism will be bluish in color, and this Mycobacterium tuberculosis or acid fast organism will stand out in color and appear as a pink organism. So we are done with the staining and direct demonstration of organism by staining. So direct demonstration of organism is very important, and this is the basically first step in identifying an organism, and also the preparing for the next step to put up the biochemical. We have to know whether it's a gram positive, gram negative organisms. For that reason, for further processing, we have to know. Then this is the first step, and next step we do is biochemical. We'll put up the biochemical according to gram stain. And next important thing, next use is that we can directly from, if it comes from the clinical sample, we can directly tell that what type of organism is there, so that they can start with an empirical therapy, whether it's a gram positive organism, whether it's a gram negative organism. Okay, so we can tell them to start with the empirical treatment of gram positive or gram negative organism or whatever it is. Next is about the classification of culture media. So we'll talk about the culture media that is used in our laboratory. The common culture media is used. So this is the classification. So we have basal media, What is basal media? Basically it consists of the basal nutrient that is required. So it's made up of peptone water. What is peptone? Peptone is basically a partially digested proteins from meat or the lean meat or from casein or from soya flour. Okay, so peptone is basically a partially digested protein. So basal media consists of peptone water, meat extract and sodium chloride, okay, electrolytes and water. Okay, so example of basal media are nutrient agar and Mueller-Hilton agar. Mueller-Hilton agar, which is used for antibiotic sensitivity test, which I'll show you later on. And next is enriched media. Okay, so basically we'll add something nutritious to the basal media to make it enriched. In case of in case of blood agar, we'll add blood into the nutrient agar. It will turn into an enriched media, or we'll add serum, or we can add egg. Okay, so example of enriched media are blood agar, where we add sheep blood agar. 5 to 10% of sheep blood agar is added to the basal media to turn it into an enriched media. And also chocolate agar. Again, chocolate agar will add the blood only, but at a different temperature. In blood agar, we'll add the blood in this nutrient agar at 40 around 44 degrees Celsius before it gets hardened up. Okay, and but in chocolate agar, we'll add the blood at 70 degrees Celsius so that the RBCs get lysis and the content of the RBC comes out, becoming and making it more nutritious than the blood agar. And another type of media is enrichment media. Okay, so what is enrichment media? Basically substance added to a liquid media to promote the growth of a bacteria or to suppress the growth of others. Okay, that is enrichment media. And examples are selenite F broth, which is a selective media for isolation of Shigella and Salmonella, and tetrahymate broth. And next is the selective media. So what we do in selective media, we add some inhibitory substance, okay, making it more selective for that particular organism. Example is XLD, that is xylose lysine deoxycholate agar, that is used for isolation of Salmonella and Shigella. Okay. Then there are indicator medias like McConkey media, clad, that is cysteine, lactose, electrolyte deficient agar, and transport media for transporting the samples, that is Carry-Blair media, Stewart's media. These are universal transport medias. So now talking about the blood agar. This is how the blood agar look like. Okay, so this consists of the basal media, okay, basal agar plus blood, 5 to 10% of sheep blood agar, and cooled it at 50. How do we prepare by adding sterile blood to the sterile nutrient agar that has been melted and cooled to 50 degrees Celsius. So blood used is 5 to 10. In chocolate agar, it is used the blood is added 70 to 75 degrees Celsius, and it is used for cultivation of fastidious bacteria and differential hemolytic reaction, like for alpha hemolysis, beta hemolysis, and gamma hemolysis. Alpha hemolysis caused by Streptococcus pneumoniae, and beta hemolysis like organisms like Staph aureus and Streptococcus pyogenes cause hemolysis to see the hemolysis. Plus this is an enriched media where most of the organisms grow in enriched media. So chocolate agar is another type of enriched media. It is more enriched than blood agar because the RBC are lysed by keeping it at higher temperature. So this is used for fastidious organisms like H. influenza, Neisseria species, and pneumococcus. We are more exacting in nutrition. For that we can use chocolate agar, which does not grow in blood agar. Okay, so composition is basically nutrient agar and the red RBC, which are RBCs, which are then lysed 75 degrees. RBCs added at 75 degrees Celsius for lysis. Okay, So these are used for fastidious organism, chocolate agar. So next is McConkey agar. McConkey agar is a differential agar and it's a semi-selective agar. Differential agar, it differentiates organism into lactose fermenting and non-lactose fermenting organism. Plus it is mildly selective because it inhibits the gram-positive organism or gram-positive commensals. So okay, so here we are using indicator neutral red. Okay, and bile salts are the selective agent which inhibits the non-intestinal bacteria or gram-positive organisms. And we also add lactose. So the lactose fermenting organism will appear as pink colonies. Why? Because if the organism is lactose producing, it will after fermentation of lactose, there will be production of acid which will change the pH. So the organism will appear pink in color, and the pH indicator here we are using is neutral red. So for some example of lactose fermenting organism are Escherichia coli and Klebsiella. And non-lactose fermenting will be colorless and take the color of the media itself. So non-lactose fermenting organism examples are Shigella, Salmonella, Proteus and Pseudomonas. So, and another enriched type of media is a blood culture bottle where we use brain heart infusion broth. Basically it's a broth. So here we are using brain heart infusion. So it is a rich source of amino acid. Here we use the calf brain and uh, heart of beef heart and calf brain is used. Okay, so they are good source of amino acids. So why we use BHI broth? Because we want the organisms to multiply in this. It's a nutritious and enriched media where basically these are used for blood culture and other sterile samples. Where the organism we suspect is very less, we want the organism to multiply. So for that reason we are using an enriched media. So here we are using this type of flat bottles, BHI broth bottle in our lab for culture of blood as well as for other sterile samples. Now another differential media is CLET, that is cysteine lactose electrolyte deficient agar. So these are basically mainly used for urine samples. Okay, so it consists of as the name of the media itself says, it consists of cysteine lactose and it is electrolyte deficient. So instead of two media using like uh, in most of the laboratories and in for most of the samples we use an enriched media like blood agar and a mildly selective media like McConkey agar. But in urine we can use just one simple clad agar instead of two agar. They will cut short the cost of the plates that we are using. So why we are using clad agar is that it is a differential media and a mildly selective media. So differential media, it will grow all the pathogen causing UTI, plus it will also inhibit the swarming of Proteus. So this media is basically used in urine for culturing urinary pathogenic organisms. So the indicator here used is bromothiamine blue. That's why the culture plate appears blue, and it differentiates bacteria into lactose fermenter and non-lactose fermenter. Okay, so if the lactose is fermented, it will turn the colony into yellow because the change in the pH. And so it is we make it electrolyte deficient because we want to inhibit the swarming of Proteus, which has the tendency to swarm and will spread out on the whole culture plate, and we are not able to isolate other organism if it is present in the sample. So that's why we use 16 lactose electrolyte deficient agar. and it also supports the growth of yeast and staphylococci, which can cause UTI. So next is the selective media. So those two were differential medias and mildly selective. Now we have a highly selective media like Lowenstein-Jensen medium. Lowenstein-Jensen medium basically it's an egg-based medium which is used for the isolation of Mycobacterium tuberculosis. So composition are malachite green. So malachite green here is the selective agent, as well as it imparts a color to the culture bottle. Okay, it imparts a green color to the culture bottle, as well as it inhibits the other organism other than Mycobacterium tuberculosis. It also consists of glycerol, which improves growth of Mycobacterium tuberculosis, and asparagine, potato starch, and coagulated egg and mineral salt solutions. So it is solidified. The basically egg white is used in this media. So that egg white is solidified by the process of by a process known as inspissation. So okay, so then it is incubated after inoculation in a slanted position with screw caps loose for at least 1 week for evaporation of excess fluid and entry of oxygen. So culture are examined weekly for growth, and we keep it for at least 6 to 8 weeks. Okay, before we say that the organism is negative. So any culture, any growth, if it's there, it will stand out as it looks like a rough buff colored. Okay, and gives a color contrast. This malachite green also gives a color contrast with the colony, so that we can easily see the new growth on this. But the main function of this, it acts as a selective agent by inhibiting other organisms. Other media used for mycobacterial culture media are solid media like egg based, I've told you, algae medium, and then these are other medias: agar based, blood based, potato based, and lipid media. So next agar is the Mueller-Hinton agar. So this is basically used in antibiotic sensitivity test. So this consists of beef infusion, casein, starch, and agar. So pH is adjusted to neutral and used for antibiotic sensitivity. This is uninoculated plate. This is with the antibiotic disc. You can see these are the antibiotic disc, and this is the growth, and this is the zone of inhibition. We measure this diameter to see the zone of inhibition, how much it has inhibited the organism, whether the organism is sensitive to this drug or resistant to this drug, and based on CLSI guideline, we will give the sensitivity of the particular organism. Okay, so thank you. This was all about today's practical. Thank you.